Okay, today we'll start the second topic, which is animal cell structure. Last lecture we talked about the overview, diversity, and basic principle and form and function. So generally here you can see the different structure between the plant cell and animal cells as viewed under the light microscope. So actually this is a diagrammatic sketch for the plant cell and animal cell. You can notice that plant cell is very well characterized by large vacuoles as well as a thick cell wall, which is lacking in the animal cell. So cell wall in the plant cell consists of cellulose, and it's not selective, so it cannot differentiate between the substance that enter and exit, and it's composed of cellulose, while it's absent in the animal cell. For the chloroplast, of course, chloroplast is in the plant cells. As you can see, the vacuoles is large and prominent in the plant cell, and these vacuoles contain permanent actually, and contain something called cell sap. And this cell sap actually is a watery solution of sugars and salts, while this is the corresponding structure in the animal cell actually is a small and temporary vesicles, and usually contain food or water. So the features as well, centrioles is only present in the animal cells. The shape is irregular in animal cells, while plant cells always the shape is regular. The size, of course, the plant cell is larger than the animal cell. Where food is stored in the plant cells, actually it is in the form of a starch granule that is stored in the chloroplast. That as well can store proteins and fats, while the food is stored in the animal cells in the form of glycogen granules in the cytoplasm, and the animal cell cannot store proteins and fats. What about the similarities between the plant and animal cell? Of course, cell membrane in the plant cell, it is present, but you can't see because you can't see under the light microscope, because normally what you can see is a cell wall, which is very thick. And it's present and it's can be visualized in the animal cells. Of course, both types of cells have nucleus, cytoplasm, ribosomes, and a lot of organelles. Let's talk about summary on the parts of the cell. So in the animal and the plant, both of them have a cytoplasm. It's kind like a jelly-like structure with 70% water, plus salts and sugars. And actually these this cytoplasm is surrounded by cell membrane and mainly have three functions: contains the cell organelles. It is actually the location of the site of the chemical reaction, as well it contains starch. For the cell membrane, it is partially permeable. It surrounds the cytoplasm as well. It prevents the cell content, of course, from escaping outside, and it controls what substance that can enter and can leave the cell. For the nucleus, which is considered the brain of the cell, it is circular oval, contain the genetic material, which is the DNA in the form of chromosomes. Of course, it is inside, the nucleus is inside the cytoplasm, and they have a lot of function we can summarize into four main points: control cell division, control cell development, control cell activities, and contains DNA, which actually is the genetic material. For the mitochondria, it is a double membrane organelle where the inner membrane of the mitochondria is folded, and it's located inside the cytoplasm, and of course it is the major site for aerobic respiration. For the ribosomes, it's either attached to endoplasmic reticulum or free in the cytoplasm. Of course, it is located inside the cytoplasm, and it is the major site for protein synthesis. What about the cell wall? The cell wall exists only in plant cells. It is tough, non-living layer, consists of cellulose, and of course it surrounds the cell membrane. That's why it's very hard to see the cell membrane. And the location, actually, it's around the outside of the plant cell. And of course it have more or little the same function of the plant cell, of the plasma membrane. It prevents plant cell from bursting and allows only water and salt to pass through. So it's freely permeable for these two materials. What about vacuole, which is only present in the plant cells? It is a fluid-filled space, as we said, contain cell sap, and it is surrounded by membrane inside the cytoplasm. It contains as well cell sap, which may contain salts, sugars, water, waste, and of course, it helps the cell, the plant cell to be firm. What about the chloroplast? Actually, it is the organ which contain a chlorophyll. It is actually the colorless blastids usually contain starch, and it is inside the cytoplasm as well. And the main function of the chloroplast is trapping light energy for the process of photosynthesis. You need to understand that chromosomes actually exist inside the nucleus, and they are very long, thin, and cannot be seen easily by electron microscope. However, when the cell is dividing, so actually you can see the chromosome because it tends to be condensed and become very short and thick and can be easily seen by a good light microscope. So if we go deeper into the ultrastructure of the plant animal cells, what's meaning of ultrastructure? That means you can see deeper inside the structure of the plant and animal cell by the using of a type of microscope which is called electron microscope. By the electron microscope, you can see the rough endoplasmic reticulum, and actually this is the organelle which carries the ribosomes, and it helps in producing and transporting and storing proteins. While the ribosomes, as we said, it's either free in the cytoplasm or bound on the surface of the endoplasmic reticulum, and hence this endoplasmic reticulum is called rough endoplasmic reticulum. In case this endoplasmic reticulum doesn't contain any ribosomes, so it will named smooth endoplasmic reticulum. Its main function, the ribosomes, is for protein synthesis. For the mitochondria, actually, it is the powerhouse of the cell. They have, as we said, outer membrane and the inner membrane, while the inner membrane is usually folded to the inside, main site for aerobic respiration. And you can see a lot of mitochondria in cells that require a lot of energy and a high rate of metabolism, like the muscle cell, the sperm cell, and nerve cell. These type of cells require a lot of energy. That's why you can see under the electron microscope that it have a lot of mitochondria, because actually mitochondria is the powerhouse of the cell, the organelle that generate a lot of ATP that is used, that the energy currency of the cell, the ATP that is used for generation energy to the cell. So here you can see that almost all cells except prokaryotes, famous example is bacteria, have mitochondria and rough endoplasmic reticulum in the cytoplasm. So you can see here a diagrammatic sketch for the liver cell. You can see the cell membrane, the rough endoplasmic reticulum, the cytoplasm between the organelles, the mitochondria is here. The endoplasmic reticulum actually appears rough because it's holding the ribosomes. You can see the mitochondria, you can see the nucleus. And what's interesting that in nucleus, there is a nuclear pore that facilitate the transport between inside the nucleus and outside to the cytoplasm and vice versa. So here actually you can see part of the liver. Actually, it's a real figure under the electron microscope that is magnified almost 20,000 times. And you can see in this magnified electron micrograph, you can see the structure of the nucleus, this whole structure, round structure is the nucleus. You can see as well the mitochondria, these dark spots is the mitochondria. You can see part of the nuclear envelope, the layer that surrounds the nucleus. You can see these small dark dots, actually the glycogen granules inside the cytoplasm. And this like enlarge the structure of a part of the liver cell. You can see it have a lot of mitochondria, which characterizing the liver cell, which needs a lot of energy because it makes a lot of functions inside our body. Here again, you can see electron micrograph of a plant cell, real figure, real micrograph of the plant cells. And you can see the chloroplast, you can see as well the rough endoplasmic reticulum, and you can see the cell wall and how it's thick. It's more obvious than the cell membrane. You can't see the cell membrane actually, because actually it is beneath the cell wall. And you can see some mitochondria there, and you can see the nucleus in the middle. Here as well, you can see electron micrograph of two liver cells together. How did we know that it is two liver cells? Because you can see two nuclei here, two nuclei separated by cell membrane, and each nuclei have its own mitochondria. Here is a more enlarged electron micrograph to show, actually it is enlarged 30,000 times, and you can see the structure of the mitochondria, and you can see the membranes of the mitochondria. So here, if you look closely to this region, you can see that there is outer membrane and there is inner membrane, and this inner membrane is folded. Here as well, you can see enlarged micrograph from the electron microscope, enlarged to 30 times. And you can see here, these are the rough endoplasmic reticulum and the ribosomes actually, which give this endoplasmic reticulum the dark color. You can see it is rough because a ribosome is attached over this endoplasmic reticulum. And of course, this dark structure, this actually a part of a mitochondria. So here this figure explains and summarizes the whole difference between the prokaryotics, the plant cell and the animal cell. So majorly you can see one of the major differences, the presence of a vacuole, a huge vacuole, chloroplast, the thick cell wall. You can see only here plasma membrane, and generally the other structure pretty the same. So majorly the difference lies in the central, the vacuole, the large vacuole again, the chloroplast, the thick cell wall, and as well the plant cell have a structure which is called the plasmodesmata, which open to the outside through the cell wall. So let's talk now about the levels of organization. So actually cells in our body are specialized. They do one particular job. They develop a distinct shape, which is different from other cells, and they do special kind of chemical reaction. So there is a somewhat called division of labor. So it is a actually the definition of division of labor, it is the specialization of cells to carry out a particular function in an organism. So you have mitochondria carry out different function than the ribosomes, than the other cell organelles as well. Even there is a specialization of cells, there is specialization in the organelles inside these cells. You have a mitochondria for a specific job, you have a ribosome for a specific job. So let's see some examples of cell that is specialized to do a specific job. One of the famous example is the ciliated cells. So you can see the cells here have cilia on top, and actually this kind of cell exists in the nose and trachea and the bronchi that passes into the lungs. And its main function actually, due to its flicking movement around, they can move mucus. Where mucus comes from? The mucus actually comes from another specialized cell, which is called goblet cell. So anything that got internalized through the nose, trachea, or bronchi, before reaching the lung, because this tract is lined by these type of ciliated cells, so the mucus can trap any dust, any bacteria going through your trachea, and then the cilia will be beating continuously upward to repel, to carry dust and the bacteria away from the lung. So this one of the important cells that exists inside our trachea and the bronchi, one of the first cell that is specialized. Then we have another cell that is specialized, which is called the root hair cell. And probably you have taken the root hair cell, this specialized kind of cell in the plant part. So actually it forms the outer layer of a young root, and mainly concerned with absorption of water and mineral salts from the soil. It is a very adapted cell. So it's long hair-like projection, and this will provide a large absorbing surface for absorbing material. Have large number of mitochondria because it needs a lot of energy for doing its job and for uptaking the minerals from the soil. The cellulose membrane, it have a very cellulose membrane. That's meaning the cell wall is much thinner than the regular plant cell to allow the short distance of diffusion from the soil to the root cell. The cell sap inside the vacuole is concentrated, very concentrated in desert plant to absorb much water, because you know that water moves from diluted regions into concentrated regions. So once the cell sap is very concentrated, that will help to the osmosis and the movement of the water from the outside to the inside of the plant. Because in a desert plant need to conserve the amount of water that is inside the plant. What about the xylem vessels? It's another specialized type of the cells that conduct water and mineral from roots to leaves and support the plant. It has two main functions, and it is characterized by strengthening by the lignin that you can see on the side walls, and actually it's a waterproof substance that thickens the wall, and you can see that the boundaries between the cells are already dead to provide a tube for the movement of the water from the roots to the leaves. So besides supporting the plant. The xylem, as we said, is made up of long xylem cells joined end to end to form a continuous fine tube that is long, thin, strong, like straw. Then we have another specialized type of cell is the palisade mesophyll cell, and it's majorly concerned with the photosynthesis and found on the upper epidermis of the leaf. It is well adapted, large number of chloroplasts to trap light energy. As we said, it is below the upper epidermis of the leaf to be exposed to the light, closely backed to absorb more light, have large surface area for gas exchange, and have large vacuole as well. Why large vacuole? This large vacuole will push the chloroplast to the edges to be more exposed to the surface to be able to trap more light. The thin walls as well, the thin cell walls of this type of specialized type palisade mesophyll cell will allow for a shorter distance for gas diffusion through the process of photosynthesis. Then we have another specialized cell, which is the nerve cell. And as you all know, the function of the nerve cell is to conduct electrical impulse along the nerve fibers. And it conveys the signal from the brain and spinal cord to different body regions. And as you can see, the cell have long fiber, which is called the axon, where the impulse travel from the cell body or the cell body of the neuron along its axon. And this axon is characterized that it's covered with a myelin sheath. And this myelin sheath actually is a fatty substance that facilitates the insulation of the axon so that the propagation of the nerve impulse along the axon will be very fast and is not interrupted. Nerve fibers are very long and connect distant far parts of a body to the central nervous system. And a lot of chemical reaction inside the nerve cell body cause this impulse to travel along the nerve fiber. Another major specialized cell in our body, which is the red blood cells. And of course, its function to carry hemoglobin, which actually a respiratory pigment that binds to oxygen and can transport the oxygen from the lung to the tissue. And it is a very well adapted cell type since it contains hemoglobin and, as we said, combined with oxygen to form another structure which is called oxyhemoglobin. And this oxyhemoglobin in lung, this oxyhemoglobin is unstable, so that means it can move, it can be transported by the blood. This cell as well is adapted by having no nucleus to provide a large space for more hemoglobin inside the red blood cells. The cells as well is biconcave disc in shape, and this as well, scientists have found this biconcave disc shape increase the surface area for diffusion. The cells are small and flexible and can squeeze, because at some point these red blood cells need to move into blood capillaries that is very narrow, so it needs to be very small and flexible. And these are the adaptation of the red blood cells. Of course, we will come to the egg cell or the ova, one of the very specialized cell that is even the egg cells are larger than the sperm cells, and they are spherical. And as you know, it is haploid and carries half the genetic information. Its function mainly in reproduction and the development into a baby when it's fertilized. It is adapted, have only 23 chromosomes, half the complement of the chromosomes, have large amount of cytoplasm containing a lot of yolk droplets that made up the proteins and made up of proteins and fats, have a jelly coat for protection and preventing for the entry of more than one sperm. The last cell type we are dealing with, specialized cell type, of course, is the sperm. And as the same as the ova or the egg, it is haploid, they have the genetic material, 23 chromosomes, mainly concerned, of course, mainly concerned with reproduction. It have at the front end of the sperm have acrosome division, and this acrosome actually responsible for secreting of enzyme that have the ability to digest the walls of the ova in order to enable this sperm to penetrate the ova. The midpiece, which is, as you can see from the figure, the midpiece, which is full of mitochondria, actually provide the energy needed for that sperm to move. And actually it is basically by providing energy to the tail or the flagella that to move and act like whip-like action to enable the sperm to move. So here you can see, before seeing the summary, do you have questions? So this table actually summarizes what we have just said. And you can see on the first column, the type of cell we have discussed, where it is found, the location we have said, the function actually that we have said for each different cell type. This is like a summary of all the specialized cell we have just said. Let's continue the levels of organization. And actually cells of plants and animals as well cannot survive by their own unless they are grouped together in a large number and made to work together. So you can see the first level of organization is the tissues. And tissues actually is a group of cells with similar structure and working together to perform a shared function. The famous example is bone tissue, nerve tissue, muscle tissue. And you can see as well that this figure will show how cells can be arranged to form cells that can form epithelium, cells that can form small tube like in kidney tubules, different cells can form the muscle tissue, different cells can form a gland like salivary glands. The second level, when the tissue is combined with different other tissue, it will form an organ. An organ actually is a structure made of group of tissue working together to perform a specific function. The examples, of course, heart is an organ, lung is an organ, intestine, brain, eyes is an organ. The next level is organ system, in which the system is composed of different organs. So a system actually is a group of organs with related function and working together to perform a body function. As you can see, heart and blood vessels made up the circulatory system, while the brain, spinal cords, and nerves make up the nervous system. So, of course, different organ system will lead to the formation of the organism. And actually, as we can say, the organism is formed by organ and system working all together to produce an independent animal or plant. So you can see here an illustration of the levels of organization. So you can see starting from cells that combine together to form tissue. You can see cells combine together to form, let's start from the organism, have a digestive system. You can see a stomach, which represents an organ. If you take part of the stomach, you can see the different tissues. And if you enlarge a part of the tissue, you can see it is consists of a different cells. So actually, this is an illustration how the liver organization work together. Another illustration you can see here as well, you can see how the system is clear. Starts from a chemical level. You can see here, starting from the chemical level of on the level of the atom that will form, for example, the molecule like a DNA. This DNA will dictate or lead to the differentiation of the cell into the peculiar or the specific cell type of the tissue. You can see now on the cellular level. You can see now the another level is cellular level, like the smooth muscle, for example. A collection or a group of these smooth muscle will lead to the tissue level. When it combines with other tissues, will form an organ level, which is the stomach. And this organ level with other organs, it will form the system or the digestive system, which eventually will lead to the organism with different other systems. Here you can see a representation for the nervous system and representation for the circulatory system, and see how the both system can form the organism. In plants as well, you can see the different system. You have a root system and you have a shoot system, and each system of these, of course, is consisting of organs. في حد عنده سؤال يا شباب على الأجزاء اللي فاتت؟ بالتالي سهلة ما فيهاش مشكلة ولا إيه؟
Yes, دكتور سهلة.
Okay. Let's talk about one of the famous examples that is structure fits function at all level. So you can see, if you look closely to, for example, we'll talk in details in coming lecture about the different systems, but you can see that this is the excretory system, and you can see when you enlarge part of the kidney, this actually the kidney, when you enlarge a part of the kidney, you can see the structure that will deal in details later on of the major unit of the kidney, which is called the nephron. And you can see how the structure fit the function, because the major function of the kidney is to do filtration of the blood and excrete with the urine all the waste product or the excess material. So actually it needs, the nephron is characterized by long, very long tube, and it is, you can see that it is in a mesh with the blood capillaries around the tubes of the nephron. So to enable the process of filtration as well of the blood. So you can see the blood is entering the nephron here at the glomerulus, it got filtered, and then while moving the filtrate of the blood through the nephron, there is a process of reabsorption as well, to get the valuable substance instead of losing this valuable material outside with the urine. So you can see that how the structure, which appear like very long tube, that enable a large surface area for the process of formation of the urine. So here, an exercise you can train at home. It is, you can see from both of these figure and try to identify examples of tissues and organ in the following both figure. You can train on these two figures at home. Now we're talking about the light microscope. Actually, light microscope, it's actually the kind of microscope you use in the lab. Actually, You need to understand that a hand lens magnifies maximum by 10 times. A good light microscope can magnify up to 1500, while the laboratory microscope, its maximum magnification power is 400 times. Usually the microscope have an eyepiece lens, which is usually 10x, and have 3 objective lenses, 4x, 10 times, 4 times, and 40 times. So the light actually can be provided by a mirror or a bulb. So nowadays, all the light microscope, the light source is a bulb. You have 2 adjustment knobs to sharpen the image and have a clear image. While the electron microscope can magnify up to 10 times million. You can prepare temporary slides, and it is very easy to prepare, but in these slides, to have some specimen on this slide to see it under the microscope, but these types of specimens can be dry out and can be ruined and cannot be stored successfully. So you need to sometimes to add the cover slip over the specimen to be able to preserve this specimen, and this will increase the time that you can store this specimen. You can use a substance called the Canada balsam that can fix the cover slip over the specimen and prevent dehydration of the specimen, and so you can keep it for many years. Here you can see a representation of the light microscope, and here you can see the eyepiece, which you have said it's a 10x. Then you have 3 objective lens, 4x and 10x and 10x and 40x, and you can see the stage where the specimen you can set, you can place the specimen there. So again, here you can see the light microscope, a diagrammatic view showing the different structure, and as well a real view of the microscope with different parts. You can see the eyepiece, you can see 3 objectives, as we said. You can see the stage where you have the specimen. You can see the stand where the light source on top, and usually it's a light bulb. And on these sides, you can see the fine adjustment and the coarse adjustment. And actually these adjustments used to focus the image, because of course everyone is different, the eyesight of everyone is different. So you have as well stage clips that used to hold in place the slide or the specimen, and you have a diaphragm that controls the amount of light through the specimen. If you need the light to be high exposure and low exposure or high amount of light to pass through the samples, or low amount of light. Of course, the 3 objective lens, it's a revolving nose piece, so you can circulate between these 3. Okay. Then we talked about the scales that we can see, that can be used to see the different structure of any organelle, even up till the atom. And what is the scale of the light microscope, and what is the scale of the electron microscope, and what the scale of the normal eye, to see which type of organelle or an atom. And here we can see the different, here we'll discuss in this slide the different types of the light microscope. So the first type of the light microscope, actually it's the bright field light microscope. And you can see you can use this bright field microscope, as you can see from the figure, you can use it to see unstained specimen. So the light passes directly through the specimen, and if the specimen is not having a natural pigment or artificially stained, the image will have little contrast, and you cannot easily differentiate between the components of the cell. And actually what is in the figure actually is the human cheek epithelial cells. And actually the scale bar denotes, or as we'll discuss later, the scale bar determine the distance. So that's mean you can see that the diameter of these cells is around 50 micrometer. And this is the first type of the light microscope, which is the bright field microscope. The second type is the bright field as well, but you can see the difference here between using a specimen that is stained specimen. You can see more contrast, the staining enhanced the contrast of the cells. And these type of cells, if you use a dye or something for staining the cells, actually you are fixing the cells. So this specimen can be preserved over a long time. But you have a downfall here, or you have a negative side here, that these cells are dead cells, because because you are using a dye to stain the cell and fix the cell, that will lead to dead cells. But in the upper case, if you use the unstained cell, you can see live cells. Here, another type of light microscope, which we normally use in cell culture, when we want to culture cells in the lab. So it is called the phase contrast microscope. And this very good microscope, it passes light through the specimen, and it uses the variation in the density of the structure of the cell, and they amplify and enhance the difference in density between the structure of the cell to give a more clear image. And it's this type of microscope, the light microscope, phase contrast, is very good to examine living unpigmented cells. So it's totally different than the bright field, and uses the theory behind the difference in density and the difference between the structure that can scatter light and absorb light to show different, like dark and gray. So you can differentiate between the different structure. Then we have the most advanced type of microscope, which is a fluorescence microscope. So in this kind of the microscope, you can see that you can label each structure inside a cell by a specific dye that can emit light with a specific color. So you need, in order to see this kind of microscope, you need the lamp of this kind of microscope to be a UV lamp. And this UV lamp will expose the UV over the different dye that will emit light in the regions of the color. And here, for example, you can see a cell that have been stained by 3 different colors. So you can see the nucleus have been stained by a blue fluorescent dye, and you can see that the cytoskeleton of the cell have been labeled with a green fluorescent dye, while you can see the structures of, for example, here, a lot of structure of the mitochondria have been stained by orange dye. So this kind of microscope, of course, you can see a live cell. You need to fix the cell, and you need to label this structure by a dye that emit fluorescence once it is exposed to UV. And actually, this type of cell is a uterine cell, type of a cell of the uterus. So it's much clearer than the other. So here we have seen 3 different types of light microscope. The types of, now we finished with the types of light microscope. We will have 2 types for electron microscope. The first one is the scanning electron microscope. And you can see from the photo, scanning electron microscope only have an image like a 3D image of the surface of the specimen. So you can see in this image, you can see this is the cilia of the tracheal cells. It looks like beating, okay? Tracheal cells in the windpipe. So only scanning electron microscope help to see the 3D structure. Normally the photos appear black and white, but using a specific software we can colorize the photos. For the transmission electron microscope, if you need to see the internal structure of these tracheal cilia, you can do a transmission electron microscope. So you can see in the photo, you can see the cilia have been cut either longitudinally or cross-section of the cilia. So basically the difference between the 2 microscopes is the scanning, it's the revealing of the outer structure of the specimen, while transmission is revealing the internal structure of the specimen. But keep Keep in mind that most of the time the photos are black and white, and the software is used to colorize the photos. Now we are going to talk about how you calculate magnification from the photo. If the specimen is viewed with a 10x eyepiece and a 40x objective lens, then the total magnification will be 400. Organelles in cells are too small to be measured in millimeter, so normally we use a smaller unit, which is a micrometer, which around 10 to the power minus 6 meter. The law of the calculating the magnification equal the size of image over the size of object. So if you consider having a mitochondria that appears in the photo, so how we can calculate the magnification? The magnification from the ruler, it will be 35 millimeter. You need to multiply with 1000 to have the unit as a micrometer. When we divide on the size of the object, the size of the object which is given is 10 micrometer. You will have the magnification around 3500. In another way, you can calculate the size of object as well, dividing the size of image over the magnification. So you have given now the given now is the magnification, which around 100,000, and you have the size of object using the ruler from the photo. So you have a photo that have an organelle that when you measure the size using the ruler, it will be 20 millimeter, and the magnification is given as well 100,000. When you use the same equation, you reach that the actual size or the size of the actual of the object is 0.2 micrometer. So the law, we can summarize the law is I equal M. I is the size of the image, A is the actual size of the object, M is the magnification. في حد عنده سؤال يا جماعة على طريقة الحساب؟ Again, magnification equal image over actual length. It is the same law. So you need to exercise these two exercises and to see how to use this information to, in first exercise, to calculate the magnification from the scale bar, and from the second size to calculate the y, the actual length of the y in micrometer. طيب يا شباب، طبعا ده قدامكم 2 exercise. You need to do this exercise as a short, a small quiz at home. Try to solve exercise 1 and exercise 2. Okay.